Okay, so today we're going to talk about the practical applications of DNA technology. So one is disease diagnosis. So figuring out if somebody has a genetic disorder. So if the sequence of a particular virus, and it doesn't even have to be a genetic disorder, it's just an infection of a virus. If a particular virus's DNA is known, so remember some viruses have a capsid and have DNA. And so if you know the sequence of the DNA, you can use PCR, which remember PCR is, go back to this, is this, where you can make many copies of the DNA. So PCR can be used to amplify a patient's blood sample and look for the small traces of the virus and looking for the viral DNA if you know the sequence. So you can do that. Another thing is gene therapy. The alteration of an individual's genes, great potential in treating disorders traced to a single defective gene. And so if we go back to and look at this picture on the other side, next page is gene therapy. This right here is the clone gene. So you would use something like PCR that I just reviewed here. And so this would be like the individual not having, let's say the individual's little r, they have a genetic disease. And so we want to insert into them the normal allele. So this would be an example of the normal allele, which means it's not present in the patient's cells. So what they do is they insert it, notice here, that's the normal gene. It says insert the RNA version of the normal allele into a retrovirus. So they take the DNA, go through transcription, make mRNA, and that's what it means by insert RNA version into a retrovirus. So the retrovirus has RNA as its genetic material. So then what they do is they take the patient's, in this case here, the patient has a blood disorder, and so gene therapy, I should mention, works best when in trying to insert it into a human cell that divides and makes other cells. Because if you put it into a cell and that cell does nothing but just sit there, it's only going to affect that cell. So it works well with blood disorders where the blood in the bone marrow, you have stem cells that can make all different kinds of cells in your blood. So if you have a blood disorder and your little r, it works well to try and replace that little r with a big R, so you in those stem cells, so that any cell that those stem cells make in your blood will have a big R and therefore alleviate the symptoms. So it's kind of like a therapeutic thing where it reduces the symptoms. So there's a disease called severe combined immunodeficiency disorder, and so these people have issues with making white blood cells. They have a disorder where it affects their immune system, and the white blood cells are part of their immune system. So the idea is to fix the stem cells in the bone marrow so that when they make the white blood cells will have the right gene. So that's what we're trying to do here. So this is like the big R gene. So you put it into your retrovirus, and then you take some of those stem cells out of the patient, and in the laboratory, it says let retrovirus infect bone marrow cells that have been removed from the patient and cultured. So it's cultured in the lab, and you let the retrovirus attack it. And so what do retroviruses do? The capsid goes into the cell. The capsid breaks down. Remember the retroviruses. They have reverse transcriptase, which, remember, reverse transcriptase can take RNA and convert it to DNA, reverse of transcription. So that RNA gets in there, it makes RNA to DNA, and then goes in. What do retroviruses do? They put their own genes into the cell, and so that right there is the big R. And now that person used to be little r, and now they have a big R, which then will cause them to make the protein that they're deficient in and take care of their symptoms. And so then they take these genetically altered bone marrow cells and inject them into the patient, and then the patient then now has stem cells that have a big R, and then they can make blood cells that also have a big R and help with their disease. All right, so that's how gene therapy works. And with that same disorder, the severe combined immunodeficiency, they actually did a trial in 2000 where they did this with 10 kids that have the disease, and actually, within the first 2 years, 9 of them actually were doing really well and had a definite improvement. But then a little bit after that, 3 of them actually developed leukemia, which is a blood cell disorder. And they think what happened is when the gene is inserted, it affected another gene that caused the blood cells to divide uncontrollably, which is what cancer is. And so that insertion caused that to happen. And so they've done some other and actually one of the kids ended up dying, by the way. And then they've done some other trials with some other diseases, not using retrovirus, but using just other viruses. And they've had some success with that. So they've done it with a blindness disorder, nervous system, and another blood disorder. But what they all have in common is they use cells that divide rapidly. All right, so there's a lot of big area of research here, a lot of promise here, but still not, we still don't have it 100%. It's hard because to know where to get the gene to insert, not to disrupt other genes. And then there's also an ethical, controversial ethical thing about whether or not it's right to even change the genes at all. Okay, so then we go back to the practical applications, and the next one is pharmaceutical production. So we've already kind of talked about this. Large amounts of protein can be produced by and human insulin is an example of this. So this is like what we just looked at in the bacteria, and getting and harnessing. Putting a human gene into bacteria, getting bacteria to make human proteins, and then we harness those proteins and give it to people who need it. Forensics, so forensic science in solving crimes. So DNA samples from blood, skin, or hair. This is in the root of the hair. Hair itself does not have DNA, but if you pull it out and you get the root bulb out of it, that has cells with DNA in it. Of alleged criminals can be compared to crime scene DNA, and that is like yesterday what I went over with this, all right, with the gel electrophoresis. Next one is paternity. We haven't talked about this yet, but it's the same kind of idea. Paternity is figuring out the father, of a baby. So DNA fingerprinting can be used. That's the gel electrophoresis process that we talked about yesterday. Can be used to determine paternity. Environmental cleanup. So I've talked about oil spills and getting bacteria, genetically engineering bacteria to make enzymes that can break down our pollutants. So I'll put here example oil spills. And then just in general, transgenic organisms. So the bacteria that do this is also a transgenic organism. This is made by introducing genes from one species into the genome of another species. So antithrombin, a blood protein in humans, that gene has been inserted into goats, and then the goats when they produce milk, that protein ends up in their milk, and then they can give to people who are deficient in antithrombin. Okay. We can also make transgenic plants, so then there's transgenic plants as well. example, we're going to talk about later on here. I'm just going to write it down here. BT corn and golden rice. I'm going to write this down here as an example, but later on here in our notes, we're going to talk about each one of those, where we inserted genes into these. So those are called transgenic organisms. Another way we'll talk about is genetically modified organisms, because that's another word for that you've probably heard. So let's flip it over here and look at some more genetic engineering in plants. So agricultural scientists have endowed a number of crop plants, so crops that we grow for food and so on, with genes for desirable traits. The TI plasmid is the most commonly used vector for introducing new genes into plant cells. This is the common name for what is used to get a gene into a cell. So example, viruses are a way, a vector. So back in the gene therapy, to get the gene into the cell, the vector was a virus, all right? So sometimes the TI plasmid is used as well. So what we have here, so the TI plasmid is, this is the plasmid itself. So they're taking this plasmid right here, and I'm taking out. So that's your TI plasmid. This is found in the bacteria. This bacteria just grows in soil. In the soil, that's where it's found naturally. And here is the big chromosome that it has, and this is the plasmid. And so the thing that's unique about this is that we can use it with plants. So if we look at here, here's the TI plasmid, and what they did is, like what we did in the lab, is use a restriction enzyme to cut the plasmid in one spot. So here's your jagged cut, and then we cut out the gene of interest, so it cuts out on both sides of it like we did in our activity, and you cut out the gene of interest, and since you use the same restriction enzymes, you can get that new gene. So let's highlight the gene purple here. So now the new gene is purple, and notice here, I'll put in, let's do yellow. This tDNA, this gene right here, is cut in half. So notice it cuts in half, so it splits in half. So this gene, the yellow gene, won't work once the new gene is put inside of it. All right, because half of it's over here and half of it's over here, and so we got the gene that we want inside the plasmid. So you have this recombinant plasmid. So then what they do is they take plant cells in the lab, and if you recall, plant cells don't take in foreign DNA. Only bacteria do. So they insert the plasmid by electroporation, where they apply a small amount of electricity that we talked about it before, that temporarily puts holes in the membrane. Allowing plasmid to enter the cell. So now it's in the cell, so that's what this picture is showing you. So now that plasmid is now in the cell, and it's what the tDNA, what happens is that part here, the tDNA, integrates in the cell's chromosomal DNA. tDNA integrates into plant cell DNA. Remember, the tDNA gene doesn't work, but the tDNA gene and your gene of interest, and that's what we're really after, is now in the plant. It is now into the plant cell. And so then what happens is that then that plant cell, the unique about plants is that those cells, plant cells, regenerate, complete plants that now have the new gene and therefore the trait, whatever that may be. So what you need to know about this and why this is a big deal is, so this TI plasmid from the soil bacteria naturally inserts its DNA into other cells. So if you put the TI plasmid into a cell, it naturally puts its DNA into the cell that it's being inserted into, their DNA. So if you put the gene of interest in there, it's a way to get a new gene into a cell. And then the other thing that's unique and why we use this with plants a lot is because cells from plants can regenerate into whole new organisms. And so you can have a select few cells and regenerate a plant. So that's what I mean by plant cells regenerate complete plants that now have the new gene. So the whole plant can come from a couple of these cells that have been genetically altered. And then that means that when they grow, they're dividing these cells, and all the cells in the plant have that particular gene in there. And so therefore the whole plant is genetically changed. That's unique with plants. Like, we can't just take a random cell from a human and grow into a whole other human. And so that was the TI plasmid using with plants as a way to genetically modify plants that we use a lot. So then, as you can imagine, there are a lot of safety and ethical questions raised by this technology. So potential benefits of genetically engineering organisms must be weighed against potential hazards of creating harmful products or procedures. So are we going to do harm by doing this? Most public concern about possible hazards centers on genetically modified organisms. You also can hear them called GMOs, and they're used as food. So when we take them into our body, people are concerned about that. So a genetically modified organism is one that has been acquired been acquired by artificial means. No, this should say has been acquired. It doesn't make any sense. Artificial means from one or more genes from another species, or even from another variety of the same species. So it can be from other species, or you can take it from another organism as well from the same species. I guess now I look back on it, the "been" doesn't need to be there. That has acquired by artificial means one or more genes from another species, or even from another variety of the same species. All right, so it does make sense as written. Some salmon have been genetically modified by addition of one or more active salmon growth hormone. So what that means is growth hormone, just like human growth hormone, causes the salmon to grow. So therefore, if they have a gene that allows them to produce more growth hormone, then they grow bigger. All right, and so a lot of salmon that is farmed, they call them salmon farms, where they're grown, not caught in the wild, but grown in pools, in buildings and things like that, are genetically modified. Most GM organisms are crop plants, though, like we talked about earlier in the video. In the U.S., most corn, soybean, and canola crops are genetically modified. And what another controversy is that they're not required to be labeled. So the grower of it does not have to tell you whether or not they've been modified or not. And some people fear that transgenic plants might pass their new genes to close relatives in nearby wild areas. So therefore, if you have a crop right here that is genetically modified, and then, you know, down the road, you've got another crop here that's not genetically modified, the fear is that wind or things like that can blow seeds, and so then you can get a mix and it's going to become part of the wild, or even if it's not a crop, even if you have forests and things like that. So you're going to, it's hard to maintain and stop it from spreading. So that's what it means by passing their new genes to close relatives in nearby wild areas. Or they also think that, some people think that the protein products of these trans genes, which means they're genes that are, you know, transferred from one organism to the next, might lead to allergic reactions, and somebody might have a bad reaction to that. So crop plants have been modified to produce their own insecticides as well. So the bacterium, it's called Bt for short, produces a protein that kills insect larvae. So this bacterium produces naturally this protein that happens to be poisonous to certain insects. And so what we've done is taken dried preparation of the Bt bacteria, and they're sold as a safe alternative to synthetic insecticides, because normally we spray chemicals on plants. And so in this case here, they have these dried bacteria, and the toxin that it produces is easily biodegradable, so that means it can break down in nature. And so some people think that that's a better alternative, because the bacteria will produce the protein that's going to kill the insects, and you're not putting a synthetic insecticide down. Genes for the toxin have been isolated, cloned, and modified and inserted into plant cells using that TI plasmid vector. So now what they've done is genetically modify plants so that the plants now have the gene that makes this protein that kills the insect larva. So they've made transgenic corn, cotton, soybeans, tomatoes, and other crops are being grown. And the use of pesticides or insecticides, pesticide or insecticide here can be kind of used the same way, is reduced because they've been genetically modified. Okay, so to produce their own insecticide. So you can imagine that this is also controversial for the same types of reasons, that there could be hazards. What about this protein that kills the insects? Is it going to affect us, and so on? Is it going to affect us in some manner? We've genetically modified crops with increased nutritional characteristics, increased or improved. So for instance, rice does not have the beta carotene. This is a vitamin that is needed to make other molecules in our body. But rice does have a precursor molecule. So when you eat rice, you're not getting any beta carotene. And there are certain areas of the world that people are deficient in beta carotene. And so, and rice is a really cheap way. It's easy to grow, and it's a really cheap way to feed people. And so therefore, the idea was, why don't we genetically modify rice to make beta carotene, and therefore give it to people that are deficient in beta carotene in some of third world countries and so on. So the genes for enzymes that synthesize beta carotene from the precursor are taken from, guess where they get it? They get it from daffodils. And they take that gene and insert it into the rice using that TI plasmid. And so now this transgenic rice makes beta carotene. Well, beta carotene is yellow, so the transgenic rice is yellow and can supply beta carotene to improve the diets of many people. And then what happens is when you eat beta carotene, it's converted to vitamin A in your body, which your body uses for lots of different purposes. And this is the golden rice, they call it. So there's different strains here. Here's regular rice, kind of enlarged so you can see it. Regular rice, white, and golden rice 1 and 2 making beta carotene. Okay, and so then they can give that to people who are deficient so that they can then make vitamin A in their body. Another example of using recombinant DNA is to get a crop to adapt to the environment. So normally plants don't live in salt water, all right? It makes so that the water diffuses out. So plants that, example plants that are salt tolerant. So then can live in saltier soils. Because if we can get plants to live in saltier soils, it increases the amount of land we can use to grow plants, and therefore we can grow plants in more areas. So what they did is they took a gene from a protein that moves sodium ions into the central vacuole, and that was isolated from a small flowering plant in Europe and was inserted into tomato plants. And so basically what it does is, remember plants, here's the cell, plant cell. Here's the central vacuole. Remember, it stores water and so on. What it does is, if sodium ions gets moved into the central vacuole, therefore it decreases the water concentration, and therefore when you have salt water on the outside, it makes it more equal, and therefore not water leaving out of the plant. And so let me show you a picture of that. So this is the small flowering plant right here that they took from the plant in Europe, that lives in salt conditions. So they isolated the gene from that plant and then put it into plants. So this is a salt tolerant tomato plant where they've inserted the gene to make them be able to live in salt water. So what this is, they're growing them. Here's the salt water down here, so it's growing. Let me just, for comparison purposes, this would be a normal tomato plant that is grown in salt water down here. You'll see that they're not doing so well. So therefore, this tomato plant is doing much, much better. And so that is a possibility that they're trying to get to increase the range in which you can grow plants by genetically modifying them. Again, going back up here, safety and ethical questions. A lot of people are still concerned about that. And so there's two very different camps, and then a lot of people right anywhere in between along the spectrum as far as what we think is okay with this. All right, and so this is where I'm going to stop with this video.